Welcome to this lesson on ATP yield from cellular respiration. When cells break down glucose, they harvest energy in the form of ATP, which stands for adenosine triphosphate. This is the energy currency of the cell. The complete oxidation of one glucose molecule produces between 36 and 38 ATP molecules. This represents the total energy harvest from cellular respiration, and understanding how this happens is crucial to grasping cellular metabolism. Let's start with the overall equation for cellular respiration. One molecule of glucose, which has the formula C6H12O6, combines with 6 molecules of oxygen. This process produces 6 molecules of carbon dioxide, 6 molecules of water, and most importantly, energy. This energy is captured and stored in the form of ATP. The complete breakdown of one glucose molecule yields between 36 and 38 ATP molecules total. This is a highly efficient energy conversion process. Cellular respiration occurs in 3 main stages, and each stage contributes differently to the total ATP yield. Glycolysis, which happens in the cytoplasm, produces a net gain of 2 ATP molecules. The Krebs cycle, also called the citric acid cycle, occurs in the mitochondrial matrix and produces 2 ATP molecules. The Electron Transport Chain, or ETC, which occurs in the inner mitochondrial membrane, produces the bulk of the ATP, between 32 and 34 molecules. As you can see, the electron transport chain is responsible for the vast majority of ATP production. Let's examine glycolysis more closely. During glycolysis, the cell actually invests 2 ATP molecules to get the process started. This is called the investment phase. However, as glucose is broken down into 2 pyruvate molecules, the pathway produces 4 ATP molecules. So we invested 2 ATP and produced 4 ATP. This gives us a net gain of 2 ATP molecules from glycolysis. It's important to remember that glycolysis is the net gain, not the gross production. The Krebs cycle runs twice for each glucose molecule because one glucose produces 2 pyruvate molecules, and each pyruvate enters the cycle separately. Each turn of the Krebs cycle produces 1 ATP molecule. Since the cycle turns twice per glucose, the total ATP production from the Krebs cycle is 2 ATP. However, the Krebs cycle's main function is not direct ATP production. It produces electron carriers NADH and FADH2, which will be used in the electron transport chain to generate much more ATP. The electron transport chain is where the majority of ATP is produced. The electron carriers NADH and FADH2 donate their electrons to the chain, which drives ATP synthesis. From the complete breakdown of one glucose, we get 10 NADH molecules. Each NADH produces approximately 2.5 ATP, giving us 25 ATP total. We also get 2 FADH2 molecules. Each FADH2 produces approximately 1.5 ATP, giving us 3 ATP. Adding these together, the electron transport chain produces 28 ATP molecules. This is the powerhouse of ATP production. You might wonder why we say 36 to 38 ATP instead of an exact number. The variation depends on which shuttle system transports NADH from glycolysis into the mitochondria. Glycolysis occurs in the cytoplasm, but the electron transport chain is in the mitochondria, so the NADH needs to be transported. The malate aspartate shuttle is more efficient and yields 38 total ATP. The glycerol 3-phosphate shuttle is slightly less efficient and yields 36 total ATP. Different cell types use different shuttles, which is why we have a range. Let's summarize the complete ATP yield. Glycolysis contributes 2 ATP. The Krebs cycle contributes 2 ATP. The electron transport chain produces 25 ATP from NADH and 3 ATP from FADH2. When we add all these together, 2 plus 2 plus 25 plus 3, we get 32 ATP. However, when we account for the different shuttle systems that transport cytoplasmic NADH into the mitochondria, the total ranges from 36 to 38 ATP per glucose molecule. This represents the complete energy harvest from cellular respiration.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.